Hi, my name is Maggie Beheler-Amass, and today I'm here to talk to you about interpretable deep learning for dynamic biophysical gene regulatory network inference. So the overall goal of dynamic gene regulatory network inference is to build predictive models. And what kind of predictive models do I mean? So the first goal that we have is to build a causal model that takes in dynamic data, and then we would like to uncover the dynamical system that's driving the cellular responses that we observe. So this would be our gene regulatory network consisting of transcription factors regulating genes. We'd also like to predict the future states of the system. So we can train our model on a cell responding to a perturbation. Let's say that we add a drug to this system. And then we would like to be able to predict the future response of that perturbation at unseen time points. Finally, we'd like to predict responses when we start our system out at a new initial state. So let's say for this first experiment, we had a healthy cell that we added a drug to, and we use that to train our model. Now we're starting out in a different initial state. So let's say a disease cell, and we add that same drug, and we'd like to be able to see if we can predict how that cell will respond over time, starting at a new initial state. And if we can achieve all of this from our model, we'll be able to potentially predict experimental outcomes in silico, creating a very powerful tool moving forward. So how can we achieve all of this from one model? Well, we can use neural ordinary differential equations to learn a predictive model for transcriptional regulation while implicitly learning biophysical parameters. So this model that I'm presenting here estimates a change in expression over time or an RNA velocity as a function of mRNA degradation, gene expression, and transcription. And this is all approximated by a neural network. So we can estimate our RNA velocity, our mRNA degradation, and our transcription directly from the model. To make this model predictive, we can integrate over our change in expression from some initial time point to a future time point using an ODE solver. And then we can add this change in expression to our initial expression and use that to then predict expression at a future unobserved time. And then finally, we can use these estimated values to infer gene regulatory networks. And so the way that the architecture of this model works is we can take expression data in at some initial time point ti and feed it into two neural network blocks, one representing the mRNA degradation, or lambda, and one representing transcription, or alpha. The transcriptional model has an interpretable deep learning architecture that infers a gene regulatory network of transcription factors regulating genes constrained by known regulatory interactions. And the weights of the entire network are constrained to be strictly positive by the ReLU activation function, and that way we can interpret the weights of the learn model as our biophysical parameter estimates. So we can combine the outputs of these two neural networks to estimate the RNA velocity at a specific time point. And then we can use our ODE solver to integrate over the RNA velocity and estimate expression at a future time point. So I tested this model on a simulated data set of 20 transcription factors regulating 200 genes with a dynamic transcriptome that created a training data set of around 10,000 cells with a dynamic expression profile for 20 minutes. And I tested the model on 2,000 unseen cells arranged into 100 trajectories to validate the model's ability to predict trajectory profiles. So the model's given some initial time point at time 0, represented in purple, and it's asked to estimate the cell's behavior for the next 20 minutes along this axis, going all the way to yellow. So this is an example trajectory of one of the 200 genes. It's gene 167. So the model's given this initial starting point at 0, and the red line is the predicted expression profile for the next 20 minutes. The black line is the mean of the actual expression values represented by the colored dots. And as you can see, the model is able to reconstruct this trajectory quite well. So next we can look at the predicted RNA velocity versus the true RNA velocity of the simulated data. The velocity here, so this dark blue line represents the actual data, the mean of the actual data, whereas the light blue line represents the predicted mRNA velocity. And we can see that the velocity is becoming slightly non-zero too early, but it's still capturing the overall shape. And then finally, we can look at the biophysical estimates. So the true values for this are in the darker colors. So decay is represented as dark red and transcription is represented as dark green. And we can see that the mRNA degradation is quite accurately predicted, while the transcription is turning on too early, which would account for why the RNA velocity is then turning on too early for this gene. But overall, the absolute error is relatively low and close to 0 for this simulated example, so we think that the model's working quite well. Finally, I'm applying my model to a real data set of single-cell yeast dynamically responding to a rapamycin perturbation, which is continuously sampled for 80 minutes. You can find the full manuscript about this data set on bioRxiv. But unlike the simulated data, this model consists of 5,700 genes and around 200 transcription factors. So it's much more complex than the model I just the system that I just presented. So here in the lower left, you have the expression profile for GAP1, which is the stress response gene. And when rapamycin is added at this dotted black line, you can see that the gene becomes active as the cells enter a starvation-like state. And the colors on the graph represent the time that the cell was sampled. So my model was given an initial starting point anywhere along the 80-minute trajectory for this data set and asked to infer expression for the next 10 minutes. And you can see that despite being a more complex system for the model to capture, the model's still learning the scaled nonlinear expression profile for GAP1, where these blue dots are the actual expression and the red is the predicted. So in conclusion, neural ODEs provide an interpretable architecture for biophysical models. Incorporating biophysical elements increases systems level understanding of regulation. The model can accurately infer dynamic regulation and predicts expression in silico, and applications to the dynamically perturbed yeast experiment show promise. So please come stop by my poster and talk to me in person. Thank you.